So you're in the life sciences, right? You're passionate about biology, but maybe you feel this pull towards the world of data, of code, of computation. If that sounds like you, then you are in exactly the right place. Today, we're kicking off a quest, really. We're going to lay out the roadmap for transforming your biology background into a seriously high-impact career in bioinformatics. And here is our map for that journey. We've broken it down into 5 key stages. First, we'll answer the call. What even is bioinformatics? Then we'll get into the foundation, building your essential toolkit. After that, the training, where you'll master the core skills. Then the really fun part, the application, taking all that theory and putting it into practice. And finally, we'll look to the horizon and figure out how to forge your own career path. All right, let's dive into stage 1, answering the call. We need to figure out what this field is all about and, why it's completely changing the game for biology. It really boils down to a huge shift in how we do science. And this slide just nails that shift perfectly. Over on the left, you've got the wet lab. This is the world you probably know. Pipettes, chemicals, petri dishes. It's absolutely crucial, but man, a single big discovery can take years of work. But on the right, you have the dry lab. This is our world. This is bioinformatics. It's where we use pure computational power to analyze biological data, and it can slash those discovery times from years down to months, or in some cases, even days. So, what is bioinformatics at its core? Well, it's this incredible fusion of different fields. The way I like to think about it is it's the engine that takes the raw, messy, just enormous amounts of data that modern biology produces. We're talking entire genomes here. And it uses computer science and stats to turn all that noise into actual, meaningful insights. It translates data into knowledge. And if you want a real-world example of its power, look no further than this. The COVID-19 mRNA vaccine was, frankly, a triumph for bioinformatics. I mean, think about this timeline. Within days of the virus being found, its entire genome was sequenced and analyzed on computers. Then, in a matter of weeks, a vaccine was designed entirely in silico inside a computer. This allowed it to hit clinical trials in months, a process that normally would have taken the better part of a decade. That is the kind of speed and impact we are talking about. Which brings us to section 2. Building your foundation. Every quest requires the right gear. And for us, that gear isn't a sword and shield. It's a very specific set of foundational knowledge and skills. And let's just get one thing straight right now. You do not need to be a coding genius from day one. Forget that idea. The whole journey starts with 2 simple, absolutely non-negotiable pillars. First, you have to have your biology fundamentals down cold. Central dogma, genetics, molecular bio, you can't analyze what you don't understand. And second, you have to learn the command line. This is your digital lab bench. It's the environment where pretty much all bioinformatics work happens. And when we talk about the command line, these tools you see here, they're like your digital scalpel and forceps. Commands like ls to just see what's in a folder, cd to move around, and something like grep to find a specific pattern in a file that's millions of lines long. These are your bread and butter. Honestly, getting comfortable with these is the first real practical step you can take on this journey. Okay, on to section 3. I like to call this the training montage part. Your foundation is secure, so now it's time to level up and really start acquiring those core programming and analytical skills that make a bioinformatician so valuable. One of the first big questions everybody asks is, which programming language should I learn, Python or R? Well, here's the breakdown. Think of Python as your general-purpose Swiss Army knife. It's amazing for building automated pipelines and connecting with AI and machine learning tools. R, on the other hand, is a specialist. It's the undisputed champion of statistical analysis and making those beautiful publication-ready graphs, especially in genomics. The truth is, you'll probably want both eventually, but this should help you pick where to start. Now, it's not enough to just know how to run the tools. You've got to understand the logic behind them. That means getting your head around core concepts, like sequence alignment. That's just how we compare 2 genes to see how they're related. Or genome assembly. That's like taking millions of tiny puzzle pieces of DNA and figuring out how to put them together to see the whole picture. And then you've got things like hidden Markov models, which sounds scary, but just think of it like being a detective trying to find a hidden gene by looking for the subtle clues it leaves behind in the DNA sequence. And where does all this amazing data even come from? Well, these public databases are basically the giant digital libraries for all of biology. You've got NCBI, which is this massive archive of pretty much all sequence data. Ensemble is fantastic for exploring genomes. UniProts is your go-to for everything about proteins. and PDB is where you find their 3D structures. Knowing your way around these is a daily skill. Okay, let's move on to Section 4, application. You've done the training, you've leveled up your skills, now it's time to put them to the test on real-world problems. This is where you really start to prove your worth. So let's walk through a classic example, analyzing data from next-generation sequencing, or NGS. It can seem super complex from the outside, but it usually follows a pretty logical flow. Step 1, you do quality control on the raw data. Is it any good? Step 2, you map those tiny DNA reads back to a reference genome to see where they came from. Step 3, you count how many reads landed on each gene. And finally, Step 4, the fun part. You use stats to figure out the biological meaning of it all. See? It's a step-by-step process. And this brings us to what might be the single most important piece of advice in this whole thing. Look, certificates are fine, but in this field, projects are what get you hired. A portfolio of real, practical projects is everything. Building something and documenting it on a site like GitHub, that's how you show an employer you can actually do the job. So where should you start? Well, here are 3 ideas for projects that will definitely get an employer's attention. You could analyze public cancer data, or maybe try clustering single-cell data to find new cell types, or you could build a tool to help predict CRISPR side effects. These aren't just homework problems, they solve real biological questions. And a little pro tip, doing projects like these forces you to deal with messy, real-world data, which is the exact experience that employers are desperate for. All right, we've made it to the final stage of our quest, the horizon. You've got the skills, you've proven you can use them, so now it's time to look ahead and choose your destination. What does a career in this field actually look like? Generally speaking, the path splits into 2 main roles. You've got the bioinformatics engineer. They're the architects. They're the ones building the heavy-duty, scalable pipelines that can process insane amounts of data. Then you have the computational biologist. They're the discoverers. They use those pipelines and develop brand new methods to ask really deep biological questions. So one builds the factory, the other uses the factory to make discoveries. So what about your educational background? Does everyone need a PhD? Well, as you can see, yeah, advanced degrees are super common. Almost half the field has a PhD. But, and this is a big but, look at the rest of the chart. There are very clear paths into this industry with a master's or even a bachelor's degree, especially if you're leaning more towards those engineer-type roles. And as we look to what's next, one thing is just screamingly obvious. AI and machine learning are not optional anymore. They're the future. We're already seeing tools like AlphaFold completely change how we do protein science. The bioinformatician of tomorrow isn't just going to analyze data. They're going to be designing, training, and interpreting these incredibly complex AI models. This is the next frontier, no question. Which really brings us to the final question, doesn't it? This fusion of AI and biology is kicking off one of the most exciting scientific revolutions of our lifetime. The roadmap is right here, the tools are available, the quest is clear. The only real question left is, are you ready to be a part of it?